One of the things we want to make sure to talk about today is the exciting translational research that has led to new treatments in spinal muscular atrophy. This is truly a new generation. I'd like to begin by talking about the first FDA-approved treatment in spinal muscular atrophy. I know, Basil, you were very involved in many of these clinical trials, and perhaps you can describe them.
I'd be happy to, Nancy. As we discussed earlier, the SMN2 gene is a major phenotypic modifier, but also has become the target of pharmaceutical research, and with the effort being trying to increase the production of full-length SMN protein from the SMN2. One of the efforts involved the development of an antisense oligonucleotide, which is a small piece of synthetic nucleic acid, which was designed to improve the splicing of the SMN2 gene. So as we said earlier, the splicing is defective, and this is the reason for the production of only about 10% of full-length protein. So there's a lot of basic science behind this development, which went on for years, to identify a particular binding site, which, if inactivated, will enhance the splicing of the SMN2 gene. They screened a large number of oligonucleotides, and one of them was found to be effective in that regard. It binds to a particular site in intron 7 next to exon 7, and by doing so, it does displace a particular protein that binds to the same site, which is a splicing repressor. And by inhibiting this particular repressor protein binding, it increases the splicing, the inclusion of exon 7 during the splicing events. So instead of having something in the range of 10%, in animals we were able to push it up to as high as 90–95%. So after a number of successful experiments in laboratory animals, they moved on to research in humans, using this piece of nucleic acid, the antisense oligonucleotide, initially known as SMNRX. And we had the initiation of clinical trials back in 2012 with a single dose given intrathecally by spinal tap. And the reason for that is that this oligonucleotide does not cross the blood-brain barrier, so it has to be given by intrathecal injection. The single injection proved to be safe, and then we moved to multiple injections with different doses, and at some point became apparent that a particular dose seemed to be effective. It specifically was 9 milligrams, and after that they moved up to 12 milligrams. So the studies continued in the beginning in an open label fashion, as we say, without having a control arm, and they showed that the medication was safe and probably effective. So it was in 2014, in August 2014, when the ENDEAR study started for type 1 babies, the severe form of SMA, in a large number of centers, not only in the US, but also internationally, which included not only a treatment arm, approximately 80 babies with SMA type 1, but also a control arm, what we called a sham procedure control arm, because we're not actually giving the medication, we just perform a sham procedure, pretending that we're doing a spinal tap. But in any case, so there was a control arm. The study lasted for approximately 13 months. And after an interim analysis in the in the summer of 2016, it became apparent that there was some degree of efficacy. In fact, the study was terminated somewhat prematurely because the data seemed to be positive. And they're positive in the sense that these babies not only were able to increase a particular score known as the CHOP-INTEND score, which measures motor function in infants, but also we noted there was acquisition of developmental milestones like head control, sitting, things like that. And of course, we noticed that the muscle strength was increased in the upper and lower extremities. In summary, there was an analysis that showed that this interim analysis showed that about 41% of the babies in the treatment arm had some kind of motor milestone response versus 0% in the sham control. That was sufficient to indicate that the medication actually was working. At the end of the study, the final analysis, in fact, the percentage of responders was 51% versus 0% in the sham control group. After this very positive finding, a decision was made to submit an application to FDA, which included also the results of the open label study that were conducted for a number of years going back to 2012. And that led to the approval of Nusinersen as the first treatment for SMA, but for all types of SMA regardless of age. So although the approval was based primarily on type 1 results, somehow FDA decided to approve it for all types of SMA, and we started treating all types of patients from very severe to less severe, from babies with severe form to babies who had who are the sitters and or walkers, and it's given intrathecally by spinal tap, 4 loading doses, then 1 dose every 4 months. We have treated a significant number of patients, not only at our site, but also other sites around the country and around the world.
And there have not only been this sham-controlled, double-blinded study in infants, but there was also one in slightly older children, the sitters, exactly, and another one that's monitoring some pre-symptomatic children, I understand. What would you say the most important lesson we've learned from these studies in comparison?
We learned that this theoretical idea that you can correct the splicing of the SMN2 actually can work in humans, but as Elizabeth said earlier, it's not actually a cure for SMA, it's only a treatment with different types of responses, from mild improvement to significant improvement, or only to stabilization of function.
And I think also comparing the outcomes and the change over time in these various treatment groups, it also appears very clear that children with the same number of copies who are treated earlier do better. And I think this is where a lot of the impetus for early identification and newborn screening is coming from.
Yes.